Any example where you have seen last reciprocal regulation? I'm asking about pathway. Glycolysis and gluconeogenesis. Fine. What is the bottleneck of that reciprocal regulation? The rate limiting step of glycolysis, PFK1, that is regulated by another bifunctional enzyme called as PFK2 and fructose 2,6-bisphosphatase. Remember? That was the bottleneck. Okay. Same like that. For glycogen metabolism also, these two processes are regulated reciprocally. Glycogen synthesis, glycogenesis, what do you expect? This is all about scenario. So if your cell is already having lot of energy, what do people will think the cell will do with the glucose? Cell is having already lot of energy. Does the cell will keep going on for glycolysis and producing energy? Not needed, na. The cell is already having lot of glucose. In that moment of time, the glucose is converted into glycogen and stored. So they are shaving down the energy. Okay? Scenario number 2, when your blood glucose level falls or your cell is less of energy, lacking of energy, what do you need? You need glucose for the production of energy. In that scenario, blood glucose level is low. So where the glucose is going to come from? Glycogen. So these two processes are reciprocally regulated. One is taking place when you are when your blood is flooded with glucose, that means in the scenario of insulin. One is getting regulated positively in the absence of glucose, when we are in fasting state, when we are starving, and as a result, the hormone responsible for regulation of this is glucagon. Yes or no? We can also add some points in this scenario. It is only insulin. It is only insulin that is anabolic in nature. Fine. There are the counterpart of insulin, the anti-insulin hormones are many. Glucagon, epinephrine, all adrenaline, epinephrine, norepinephrine, thyroxine. These all play a very important role in anti to insulin actions. So insulin is the only one barrier fighting against all of them. Okay? Remaining all of them, glucagon, epinephrine, norepinephrine, thyroxine, they all are catabolic by nature and they are responsible to maintain blood glucose homeostasis. Okay? Whereas insulin plays important role in preserving the glucose provided for future time. So that whenever the there is a downfall of glucose level in the blood, you can use this component. Same like stands for gluconeogenesis. Achha, aur ek cheez bolu. Interesting. When we talk about glycolysis and gluconeogenesis, and when we talk about insulin glucagon, okay, in an overall scenario, what we see that insulin is an anabolic hormone. That insulin is responsible for forming something, utilizing the glucose. Okay, but glucagon itself is a catabolic hormone, which allows the breakdown of something and provides energy or glucose, whatever. Both of them carries one exception. Can you name that? In terms of pathways. Insulin regulates certain pathways which are anabolic in nature. See, fatty acid synthesis, cholesterol biosynthesis, triacylglycerol synthesis. These all are supported down by glycogen synthesis. These all are supported down by insulin, activated down by insulin. Okay? Whereas glucagon, it activates process like beta oxidation, glycogenolysis. Is that clear? So it is catabolic by nature. Is there any exception in terms of pathways for insulin and glucagon? Already taught. Is there any exception? What I'm saying, insulin favors anabolic reactions, pathways. Glucagon favors catabolic pathways. Yes or no? Clear? Is there any exception to this point? Yes. Insulin favors glycolysis or gluconeogenesis? Insulin favors glycolysis or gluconeogenesis? Still confusion. Somebody is saying gluconeogenesis. Glycolysis is favored in the presence of insulin. ठीक है? PFK1 gets activated. Hormonal regulation याद करो. On other scenario, gluconeogenesis is activated or favored by glucagon. Insulin favors anabolic pathway, I told you. Glucagon favors catabolic pathway, I told you. Is glycolysis an anabolic pathway? Are you forming something? Is something new coming out of glycolysis? It is a breakdown of glucose. So it's a catabolic pathway. So the only exception stands for insulin in terms of their metabolic regulations and metabolic pathways is that glycolysis. Same stands for gluconeogenesis. The only exception is gluconeogenesis in presence of glucagon, because glucagon plays an important role in catabolism, not anabolism. Okay? And gluconeogenesis is a process of anabolism. It is forming glucose from some other non-carbohydrate precursors. That's why we need energy. Gluconeogenesis produces energy or it needs energy? Needs energy. It should be anabolic by nature. ठीक है? Confuse मत करना कभी इन चीजों में. Okay. What is glycogen? All of you must know. What is glycogen? Stored form of energy in animals, people. Okay. What is the structure of glycogen then? Is it a linear structure? No. It is a branched structure. Heavily branched or moderately branched? Heavily branched. Very good. The excess number of branching makes it more insoluble or soluble? फिर से बोलो. How many of you say that increase in branching points makes the glycogen molecule less soluble? If you increase the number of branching points, the glycogen becomes less soluble. How many of you? No. If you increase the branching point, glycogen molecules become more soluble. Raise your hand. All of you? अरे सब बीच में खड़े हो तुम लोग, एकदम सेंटर लाइन में, ना इधर मुड़ पा रहे हो ना उधर जा पा रहे. It makes it more soluble. Yes. It makes it more soluble. And because of the solubility, it is easily stored in a small packets. It can be easily stored. Okay. Point number 2. Storage and location. What do you think glycogen is present? Where? Is it present in all the cells? Is it present in all the cells? Yes or no? No. If I say yes, then will you agree me? Are you going to agree with me? Yes, it is present in most of the cells, but in a very definite manner, in a very small manner, concentration. Okay? But the majority of the glycogen is specifically stored in liver and skeletal muscle. Okay? To some extent in kidneys also. Okay? Where do you think the glycogen storage will be more? In liver or skeletal muscle? Because liver is the largest organ, isn't it? Arey pagalo. Skeletal muscle or liver? Skeletal muscle, yes, of course. Okay? Do you know the concentration? The skeletal muscle contains around 400 gram of glycogen storage. That contributes to only 1 to 10% weight of your skeletal muscle. The total weight of your body provided by the skeletal muscle, उसमें से 1 to 2% निकाल दो. That will be equal to your weight of glycogen stored in your skeletal muscle. Okay? If you talk about a well-feeded, healthy liver, it stores around 100 grams of glycogen that contributes to 10% weight of your healthy liver. Important, ये कभी कभी आता है क्वेश्चन, नीट पीजी वगैरह में आते हैं ये क्वेश्चन. ठीक है? Remember this. What do you think the enzymes of this process, both the process, are present in? These enzymes are localized in cytoplasm. Okay? So basically, glycogenolysis or glycogen breakdown, both the process are occurring only in cytoplasm. Yes or no? They are cytoplasmic by nature. Interestingly, whenever we talk about storage of glycogen, when we store glycogen, when we are, let us say like when we are synthesizing glycogen or when we are degrading glycogen, these all are occurring inside small vacuoles. Some secretory vesicles, okay, some vesicles, small vacuoles. And interestingly, the enzymes responsible for synthesis of both for the synthesis of glycogen as well as degradation of glycogen are present in that vacuole only. Are you getting my point? The glycogen storage, if you talk, is present in small vacuoles inside, inside the cytoplasm. ऐसे छोटे छोटे vacuoles हैं, उसके अंदर glycogen stored है, ठीक है? And interestingly, the enzymes responsible for glycogenesis as well as glycogenolysis are present in these vacuoles only. Am I clear? So, the control of this regulation is very important. Otherwise, there will be either there will be excessive storage or there will be excessive degradation. So the control of this hormone is these enzymes provided by hormones is very important, and that is provided at hormonal regulation. That through with the help of covalent modification. We'll talk in that later part. Okay, about that. Okay. If we talk about that glycogen supports our glucose need or body need, that for that reason only it has been made up. Liver breaks down the glycogen and provides the glucose for blood. Yes or no? So basically, can we say that the liver glycogen is required to maintain glucose homeostasis? Yes or no? Can we say? We have got a general, range stating it to be 70 to 100 mg per dl of blood glucose level, fasting. Generalized, okay. According to ADA, American Diabetes Association. Fine. So, when the blood glucose level falls below 70, in overall I am saying that that is not the right criteria to convey the message. But in general I am saying, if it falls below 70, then what should be the role of liver? It should undergo glycogenolysis and produces glucose that goes into the blood and makes the blood, again the concentration of glucose again in the blood rises. ये criteria होना चाहिए, ठीक है? And in the same time, what your skeletal muscle is doing, the glycogen stored in the skeletal muscle is for what? For itself, for its own purpose. What is the purpose of glucose in skeletal muscle? For production of ATP, because we require it for contraction of muscles and all, energy is required. Fine. So what is the role of glycogen that is stored in skeletal muscle? Point, just let me know. Point number 1. Is it going to provide glucose to the blood? No. Remember this point. Skeletal muscle or the glycogen stored in skeletal muscle is not responsible for increasing the blood glucose level. Remember that. In other way, you can remember it by that skeletal muscle is actually matlabi in terms of glycogen. Selfish. Is that clear? Am I clear? So, muscle storing glycogen can only utilize the glycogen for its own purpose of energy, but cannot increase the blood glucose level. यहाँ पे question आता है कि why? I will answer this question at a later part. Remember this, okay? Even interestingly, liver glycogen responds to fall in glucose level. In a short period of fasting also, the liver glycogen will get depleted. But muscle glycogen does not respond to short periods of fasting. One day, 2 day fasting होता है ना, करते हैं ना हम लोग. In that scenario, the muscle glycogen is generally not depleted, but the liver gone. Liver goes out. The liver glycogen is depleted totally. But muscle glycogen takes around a longer period of fasting, for a week kind of thing, to get depleted. And one important thing, who decides that? How it is decided? Okay, which cell of the body comes to know, or which platform of your body comes to know that okay, this has been 2 days. No, don't use the muscle glycogen. Use something else. Okay, it has now been 1 week. Chalo, deplete karo muscle glycogen. Who decides it? It's still under research. There are so many things left to know about our body. Okay, it is still under research. People are still not clear about this. The point that who clarifies, who instructs the brain or who instructs our body, who instructs our tissues to not or utilize the glycogen whenever in need. It is still not known. Okay. Okay. So this is general introduction. Moving towards process. Which one you want me to start with? तोड़ने बहुत अच्छा लगता है क्या तुम लोग को? पहले बनाओ तो सही. क्या बनाना है या तोड़ना है? लड़का लोग क्या बोलेगा? तोड़ना है सर. है? बनाना है कि तोड़ना है? बनाना है. मेरे पास नहीं है बनाना. तुम्हारे पास है तो दो. भूख लगा. ग्लाइकोजेनेसिस या ग्लाइकोजेनोलाइसिस? जेनेसिस. देखो. Why I ask this question? Because many of the books, standard books, they have written glycogenolysis first, and then they move to glycogenesis. Don't do that. Don't do that mistake. First, you should go through glycogenesis, synthesis, then you go with that. There is a reason why. You will come to know. First of all, whenever you talk about these two pathways, I suggest you to write down or remember the names of the enzyme that you are going to look for, because if you are not comfortable with the enzymes, this process is going to be tough. Okay. First enzyme that participates is called as phosphoglucomutase. Okay, first. Second enzyme that plays a role is called as UDP-glucose pyrophosphorylase. What is the role of phosphorylase enzyme? Can anyone say? What is the role of phosphorylase? Pyro को छोड़ दो, भूल जाओ. Pyro, why I have added pyro? Forget about it. What is the role of phosphorylase? There are three classes of enzymes. Again, some hint I am giving. Some knowledge I am sharing with you. Kinase, phosphatase, phosphorylase. Anyone? Kinase, you know, it is responsible for the transfer of phosphate group. Shart hai ki the donor should be an ATP, GTP, ITP. Okay? Phosphatase is a hydrolyzing enzyme. That means it adds or incorporates water and breaks a phosphate group. Removes the phosphate in the form of PI. Yes or no? ठीक है? Then what is phosphorylase? Superb. It is the role of phosphorylase is both of the phosphatase and kinases. It adds as well as removes also. Fine. If it is adding something, for example, UTP is added, UTP goes into the reaction, UDP, one phosphate is coming out, UDP is added to something and one phosphate goes out. So performing both the action. Okay. Next comes glycogen synthase. Okay. And the last enzyme comes as branching enzyme. I have written it like branching enzyme, but you are not supposed to remember it as branching enzyme. You should remember that with the help of name itself. And the name is, अभी तुमको ये tough लग रहा होगा name देखो. It is named as amylo alpha 142 alpha 16 trans glycosidase. This is the name of this branching enzyme. Amylo 14216 trans glycosidase. ठीक है? अभी समझ में नहीं आएगा तुमको ये एंजाइम का नेम क्या है. Am I clear till here? Can you all remember the names? Can you all remember it? Can you say me the first enzyme? Phosphoglucomutase, UDP-glucose, pyrophosphorylase, and glycogen synthase, and branching enzyme that is called as amylo 1416 trans-glycosylase. Any doubt? Any doubt? Chalo. Let us now synthesize a molecule of glucose. Fine? The first precursor for glycogen is glucose 6-phosphate. Where is this glucose 6-phosphate coming from? Where is this glucose 6-phosphate in the cytoplasm coming from? Glucose. Remember the first step of glycolysis? I told you na, it is a preparatory step. Glucose is phosphorylated at the 6th carbon just to keep the glucose inside the cytoplasm so that it cannot leave out of the cell. Yes or no? I have told you. Fine. I have also told you one more point, that formation of glucose 6-phosphate is not the dedicated step for glycolysis. It is not the committed step of glycolysis. Isn't it? I have told you that the committed step for glycolysis is the formation of fructose 1,6-base phosphate. So fructose 6-phosphate converts into fructose 1,6-base phosphate. It is dedicated for glycolysis. It cannot go anywhere else. Yes or no? But the formation of glucose 6-phosphate, I have told you that glucose 6-phosphate can go to other pathways like HMP shunt, like glycogen synthesis. It depends on the cellular need and availability. Okay? Now you imagine what we are doing, synthesizing the glycogen. Okay, your cell is already having lot of glucose, lot of energy. Do we need to go for glycolysis? No. But we need to lower down the glucose level. So for lowering down the glucose level, what we will do? What the cell is going to do? Convert or shuffling the glucose 6-phosphate from glycolysis to glycogen synthesis, dependent on the availability of energy and glucose. Clear? The first step is conversion of glucose 6-phosphate by an enzyme called as phosphoglucomutase. Enzyme number 1. Okay, and it converts glucose 6-phosphate into glucose 1-phosphate. What is happening? Simple. The 6-phosphate group is transferred to the first carbon. That's a terminal first carbon. Fine. Reaction number 2. Enzyme name is UDP-glucose pyrophosphorylase. Okay. What is happening? As I told you, the action of phosphorylase. A UTP molecule donates its UDP, to glucose, and in return to that, because it's a phosphorylase, so it will donate and will remove also. He has donated the UDP 2-phosphate group, uridine, from uridine triphosphate. So 1-phosphate group has gone out. Yes or no? Then where is that other phosphate group coming from? I have written it as pyrophosphate. This is why the enzyme name is called as pyrophosphorylase. Why this PPI? Where is this? 1-phosphate group is coming from UTP, I understood. Where is that other phosphate group coming from? There are 2 inorganic phosphates coming out of the reaction. One is coming from this. It was having 3 phosphate. 1, 2, 3. One was removed. UDP left. UDP was transferred. See. Where is the one more phosphate group coming from? Yes. This is the phosphate group that got removed. Is that clear? And this structure is called as pyrophosphate. This is a very unstable structure. Wherever you see this kind of reaction where PPI is formed, this is a very unstable structure. Immediately it has to be broken down by an enzyme called as pyrophosphatase. Pyrophosphatase, by converting them into 2 molecules of PI. And PI is acceptable form, is a stable form. Is that clear? Am I clear? Okay. Why do we need UDP glucose? Why do we need to form UDP glucose? Because glucose is less polar. It cannot directly move towards the formation of glycogen, it needs a carrier. It needs someone to hold the hand of glucose and take him towards the formation of glycogen. Glucose itself cannot reach that area. Okay? Point number 2. The next reaction that you are going to see is provided by glycogen synthase. What we understand by synthase? We are joining two things. Yes or no? So if glucose goes alone and ask the samne wala person or the component, okay, join me. Allow me to join you guys. But the glucose will not be having energy. Is the joining process possible? Is the joining process possible? No. The joining process is not possible. So point number 2 is, for the joining, for the attachment of glucose molecule provided to a primer that we are going to discuss later, primer, we require a transporter, a carrier. Someone has to carry the glucose. Point number 2, for the energy that is going to require for the addition of glucose. Okay? The breakdown of this bond will provide the energy. Point number 3, the direction of this conversion of 2 ppi into pi gives a hint that okay, from the next, for the energy provided for the next reaction will be provided by UDP-glucose or by the breakdown of pyrophosphate to 2pi. Forward direction. Am I clear? Okay. Next reaction is provided by glycogenin. Have I written any name of like glycogenin and all? Where is that coming from then? What is glycogenin? Glycogenin is a precursor. See, whenever we are our liver cells or the skeletal muscles are breaking down glycogen for their utilization, some amount of glycogen is left. You cannot complete. Again, one more question. That who decides that this much amount of glycogen have to be stored, we have to keep it inside the cells. We cannot deplete it completely. Some amount has to be there. Who decides it? Still under research. It's still not clear. Okay? So some amount of glycogen or some protein carbohydrate moiety, proteoglycan has to be there, or glycoprotein has to be there. And that glycoprotein is called as glycogenin. Okay? This is a glycogenin, remember. Glycogenin. Basically, glycogenin is nothing but a protein with a structure of some branches, short branches of glycogen. Short branches of glycogen. Acting as a primer. RNA primer? Remember? Is it a long strand? No, it was short. Fine. That allows the addition of deoxynucleotides. That accepts. Same role is going to be played by glycogenin, which is a protein, which is a glycoprotein having short branches of glycogen attached to the protein. Yes or no? This, as I told you, UDP attachment to glucose was done just to carry the glucose to the glycogenin, but the glycogenin cannot accept the glucose from UDP glucose directly. The UDP glucose cannot add directly. Okay? It requires some primers, and these primers, the short chain of glycogens, are very short, very small by nature, all showing up their non-reducing ends, but very small in range. In that scenario, some, like for example, this is what the advancing branch, let us say, this is what the glycogenin, the protein part, this is what the glucose moiety is present. Okay, let us say. Okay, small chain. Fine. Now, the glycogenin is it is a protein, but it functions as an enzyme. Remember this question has came as I'm forgetting this year, maybe 2021, because on 2019 only it got this theory was proposed that glycogenin also act as an enzyme. It is a protein, but it also acts as a enzyme also. Okay. In 20 or 21, I think it came in NEET PG. NEET PG में ये question पूछ रहे हैं. Okay, about the innovations actually. See, what is glycogenin doing? Glycogenin is basically adding 4 molecules. How many? 4 molecules of glucose from UDP-glucose. As a result, UDP goes out. This UDP can convert back into UDP and can participate in this reaction. Fine. So first 4 molecules in a linear strand is added upon by glycogenin. The action of this protein is limited for the addition of only 4 linear branches, linear glucose molecules. Limited. उससे ज़्यादा नहीं कर पाएगा. क्षमता ही नहीं है उसका. ठीक है? Now comes the role of glycogen synthase, where glycogen synthase now adds glucose again by removing UDP till the branch advances to more than 8 to 12 glucose residues. Let us say this is the branching point. ये जो points अभी मैं बोल रहा हूँ ना, यही points important है इसको याद रखने के लिए. Glycogenin can add only 4, max to max 4 molecules of glucose in linear way. ठीक है? When it is done, it comes the role of glycogen synthase, which will add glucose in 1,4 linkage, alpha 1,4 linkage. ठीक है? Till the moment the size of linear chains of glycogen reaches up to 8 to 14 glucose residues. मेरा बात समझे? Till the size reaches to 8 to 14. The moment the glycogen becomes bigger, and the action of glycogen synthase halts when the length of the glycogen molecule or the 1,4 linear polymer of glucose reaches up to 8 to 14 glycosyl residues. Yes or no? Am I clear? ठीक है? The moment action of glycogen synthase halts, the important point comes for branching enzyme. Now what branching enzyme is going to do? See. Now comes branching enzyme. What is the name of branching enzyme? Amylo 1,4,2,1,6 transglycosylase. नाम ही suggest कर रहा है देखो. What this enzyme is going to do? From an advancing chain in a linear way, advancing chain जो कि 8 से 14 glycosyl residues बड़ा हो चुका है, what it will do? It will remove a block of 6 to 8. How many? 6 to 8. बना नहीं पाया हो? 6 to 8 imagine करना. It will remove a block of 6 to 8 glucose residues present in linear form, and they are going to transfer them to the above branch, providing a 1,6 linkage. कोई doubt है यहाँ पे? Is there any doubt? What I said? The glycogen was advancing linear. ठीक है? It reaches a length of 8 to 14 glycosyl residues. Action of glycogen synthase halts. Now comes branching enzyme. What branching enzyme does? Amylo 1,4. ये है 1,4 linkage. Linear linkage है. Branching point नहीं है. Linear linkage. ठीक है? This amylo 1,4 transglycosylase, what it will do? It will transfer a block of 4 or more than 4 molecules of glycosyl residues in linear form, in 1:4 form, and it will transfer it to 1,6 linkage. देखो, यही सारे हैं. Branching point हो गया. This is what is the linkage? 1:6. Yes or no? ठीक है? Name suggests amylose. ठीक है? Branching only linear, no branching point. Amylo only branching. Sorry, only linear, no branching point. Removing the alpha 1,4 glycosyl residues and adding upon over a top of it and forming 1,6 linkage. This is the 1,6 linkage, you know. This is 1,6. This is 1,4. This is 1,6. This is the role of this enzyme. Yes or no? Fine. Again, glycogen synthase comes and starts adding glucose in both the chains linear wise. फिर वो 8 से 14 पहुंच जाएगा, ऐड करते 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 8 से 14 रेसिड्यू जैसे ही बढ़ जाएगा, फिर ब्रांचिंग एंजाइम विल कम इनटू प्ले. What it will do? It will add one more amylose structure, linear structure, and transfer it to one more 1,6 branch. So this is how the branching of glycogen keeps on going. Am I clear to all of you? Am I clear? Good. Now, this is how glycogen forms. Okay. Next, moving towards glycogenolysis. What is the process of studying this? Which word? Let it go. Achha, fine. Okay. Sorry. It is there in that video, no? You can get it. Don't worry. Now we are going towards glycogenolysis. Breakdown. Okay. Listen. See all of you. What are the enzymes? We require the first enzyme, glycogen phosphorylase. Glycogen phosphorylase. Yes or no? ठीक है? Next, debranching enzyme. Now, important point is debranching enzyme is a bifunctional enzyme. That means it has got two different domains. The first domain is responsible for removal of 1,4 linkages, that means linear linkages. Another is responsible for removal of 1,6 linkage. ठीक है? What we call them as alpha 1,4 alpha 1,6 glucosidase, alpha 1,4 glucan transferase, alpha 1,4 glucan transferase. It is called as glucosidase. It's a bifunctional enzyme. Okay, both of them will be allowing the debranching of glycogen. Out of that, specifically alpha 1,4 glucosidase will responsible for the removal of glucose unit. Okay. Next enzyme comes as phosphoglucomutase. Have you seen this enzyme for glycogen synthesis? Yes or no? This enzyme was there, no? Yes or no? Yeah. Phosphoglucomutase is a non-regulating enzyme and that participates in the case of both the process. Let that be glycogen synthesis, let that be glycogen breakdown. Phosphoglucomutase is a non-regulating enzyme that participates in both the pathways. Okay. The last and the very important enzyme, glucose 6-phosphatase. Okay. Am I clear to all of you? So, here comes the process. Here comes the process. You have a molecule of glycogen. Let us say this is the molecule of glycogen. This is where you have a branching point. This is 1,6. These are all 1,4. Okay. These are the non-reducing ends. The first proposed enzyme that comes into play is glycogen phosphorylase. What is glycogen phosphorylase? It is a regulating enzyme. What is the role of it? Glycogen phosphorylase breaks down 1,4 linkages of glycosyl residues, but the action limits, ध्यान से देखिएगा, the action limits or halts the action of what? The glycogen phosphorylase. The action of glycogen phosphorylase halts 4 glycosyl residues before the branching point. What did I say? Can you repeat? What is the function of glycogen phosphorylase? It is going to break down the glucose present in 1,4 linkage, only linear linkage into certain thing. Okay, let me say the product is glucose 1-phosphate. Okay, is that clear? But the action of glycogen phosphorylase is going to halt at a point which is 4 glycosyl residues away from the branching point. This is the branching point beta. उससे पहले 4 glycosyl residues पहले तक वो काम करेगा कौन? Glycogen phosphorylase. उसके बाद जैसे ही 4 तक आ जाएगा, it will halt. The action of glycogen phosphorylase will halt. It will stop working because glycogen phosphorylase cannot break down 1,6 linkage. Am I clear? For that, what you need? You need debranching enzyme. So what we have got 2 classes, 1,4 alpha 1,4 glucan transferase, another is 1,6 glucosidase. Okay. So now your structure is appearing like this. See. Sorry. Your structure is appearing like this. Short ho gaya length. It has been shortened. Yes or no? The first enzyme that plays a role is called as alpha 1,4 glucan transferase. They could just go through the name. You'll come to know what is happening. This enzyme is going to transfer a block of 3 glycosyl residues in linear form. This enzyme is going to transfer a block of 3 glycosyl residues in a linear form. Linear form बोले तो alpha 1,4, इसलिए इसका नाम alpha 1,4 है. And is going to transfer them and join them in linear form in the lower branch. Yes or no? Am I clear? But it will not interfere with the 1,6 linkage. That's why the name is alpha 1,4 glucan transferase. Yes or no? Is that clear? Now the scenario or the structure looks like this. ऐसा दिख रहा है? You have only one left. This is what. Now the enzyme comes is alpha 1,6 glucosidase. Simple, it will remove the 1,6 linkage and a free glucose molecule is released. Yes or no? An important concept about this specific step is, can we say that the number of free glucose molecule released during the degradation of glycogen is equal to the number of branches present in glycogen molecule? Arey, all of you. Can we say? Raise your hand those who, convey, they are conveyed with my statement. Can we say that the number of free glucose molecule released during the process of glycogen degradation or glycogenolysis is equal to the number of branching points present in the structure of glycogen? So how many of you would says like yes? How many of you say no? तो बाकी लोग कहाँ जाते हो तुम लोग? कौन सा ऐसा पार्क है जहाँ घूमने निकल जाते हो अचानक से क्लास के बीच में? एक तो यस में रहो या तो नो में रहो. कॉम्प्लिकेटेड वाला स्टेटस अपने लाइफ में मत रखो यार. इट्स कॉम्प्लिकेटेड. उसको अलग सेगमेंट में रखो उसको बोलने के लिए. फेसबुक के स्टेटस में दो, इंस्टा के स्टेटस में दो. स्कॉर्पियन बहुत इंटरेस्टिंग ये स्टोरी है. इंस्टा का स्टोरी में लिखा हुआ है, देख रहा था मैं. स्कॉर्पियन, we can say number of free glucose molecule released during the process of glycogen degradation is equal to the number of branching points provided in the process, in the structure of glycogen. We can say, okay, this free glucose can go out into the blood supply and it can ultimately be used for the process of energy or for increasing the blood glucose level. Okay, but now the point is not it, the process is not yet complete, because we have glucose 1-phosphate, so many glucose 1-phosphates. What the structure will be looking like now? ऐसा दिखेगा पूरा linear. Next, again glycogen phosphorylase will come act on it and will provide many more molecule of glucose 1-phosphate. So what you will see, that after the degradation of glycogen is completed, there are a lot of glucose 1-phosphate molecules inside the cytoplasm, and glucose 1-phosphate molecule is not going to be accepted by any other pathways. ठीक है, कहीं पे देखिए ग्लूकोज 1-phosphate is entering as a substrate? No. ठीक है, so that's why the next step that is required is to convert these all molecule of glucose 1-phosphate into a component called as glucose 6-phosphate, and that is provided by enzyme called as phosphoglucomutase. Phosphoglucomutase. ठीक है, फिर भी there is one last enzyme left, glucose 6-phosphatase. What it is going to do? Here comes the tragedy. The tragedy is what happens for the glycogen if the glycogenolysis is occurring in liver. What happens with the glycogen of muscle if glycogenolysis is occurring? ठीक है, one question for all of you, एकदम फटाक से आंसर आना चाहिए. Glucose 6-phosphatase is an enzyme. Is this the first time you are seeing this enzyme? Yes or no? Where have you seen this enzyme? Glucogenesis? नहीं देखी हो? Glycolysis में enzyme था? सही है भाई? Where have you seen? Glucogenesis. Glucogenesis is the last enzyme. Remember, glucose 6-phosphate converts into glucose by the enzyme glucose 6-phosphatase. ठीक है? Point number 2. If you have seen this enzyme in a process called as gluconeogenesis, then what should be the localization of this enzyme? Tissue level. What is the localization of this enzyme? What should be the answer? Liver. ठीक है? So this enzyme is present only in liver. Muscle में नहीं है. There is no glucose 6-phosphatase in the skeletal muscle. ठीक है? Am I clear? So as a result of that, the glucose 6-phosphate formed in the liver will convert into glucose, and the glucose can go into blood and increase the blood glucose level. But due to the deficiency, inborn deficiency, भगवान बना के दिया ही नहीं, glucose 6-phosphatase in muscle, due to deficiency of glucose 6-phosphatase in muscle, glucose 6-phosphate cannot convert into glucose. Yes or no? Fine. As a result, the glucose 6-phosphate goes into glycolysis for muscles and provides ATP that can only be used by muscle. Yes or no? Can anyone out of here raise their hand and say me that if glucose 6-phosphate is getting formed in the muscle, why it cannot go out of the cell? It is getting formed. If glucose 6-phosphate is getting formed, it can go out of the cell, na? It can go out of the cell of a skeletal muscle, it can come into the blood, it can travel from brain to, tip of the toe. Phosphorylated components cannot leave the cell because they don't have specific transporters. Remember? So glucose 6-phosphate cannot go out of the cell anyhow because the cell membrane has got no transporters for the phosphorylated components. As a result of that, the glucose 6-phosphate has to stay inside the muscle only. Is that clear? ठीक है? Last point. There is a disease called as a disease with a very high incidence rate. Next class में हम लोग पढ़ेंगे उसको. That is called as von Gerkes disease. ठीक है? ध्यान से सुनना, मैं कुछ पूछने वाला हूँ. देखता हूँ कितना स्मार्ट हो तुम लोग. Von Gerkes disease. Von Gerkes. Okay. हाँ, von Gerkes भी बोलते हैं कहीं पे. It's fine. Okay. That is due to the deficiency of glucose 6 phosphatase. If there is a inborn deficiency of glucose 6 phosphatase, fine, this disease occurs. So the most common effect of von Goetz disease should be happening in liver or skeletal muscle. Skeletal muscle में glucose 6 phosphatase होता ही नहीं है, तो deficiency कहाँ से होगी उसकी? It is present only in liver. So the major affected part or organ in von Goetz disease is liver.